going to talk about the role of artificial intelligence for finding and using biomarkers of response to drugs and radiotherapy. So what is the first step when using AI to find a new biomarker?
So the first step is to think about what kind of samples you want to use to find your biomarkers. Is it going to be a tissue sample? Is it going to be a blood sample? Most biomarkers are generated from tissue samples from the tumor itself, so we can go very deep in understanding what makes that tumor very unique. So you generate those data using different sequencing technologies. Could be RNA-seq, DNA sequencing, epigenomics like methylation or ATAC-seq. From this very, very complex data, that's where you use AI to look at the patterns that associate what you see in the tumor with the response of the patient to a given therapy.
And what kind of AI do you use for that?
So AI is a broad term for a lot of different techniques. Some are more traditional machine learning. Some are really complex like deep neural networks. And we use really a big portfolio of those techniques. But the recent advances in AI applied for biomarker discovery really come from the deep neural network. So we try to kind of simulate the way the brain works to understand how those patterns, how those gene expressions or mutation or other features can be connected to each other to make the most accurate predictions.
How works the evaluation and clinical validation of a potential new biomarker?
You actually point out the biggest gap in AI for biomarker discovery right now. We generate a lot of data, not only during clinical trials, but also in the lab to try to understand how can we build those predictors of drug response. The problem is that in clinical trials, it's very expensive to generate large amounts of data. A clinical trial usually costs millions of dollars to organize, plus all the resources needed to generate the data. So we usually use preclinical models in the lab, cancer cell lines, tumors in mice, to basically challenge those cancer cells with different lines of therapies, different kinds of therapies in parallel. It is a very rich information. The problem is tumors growing in a lab are not really exactly behaving the same way than tumors in a patient. So there is this big gap in translation between the data we use to build those biomarkers and predictors of drug response and the actual applications in patients. And so validation of those predictors is a big step where we actually need clinical information to validate the predictions. And so now with our many initiatives where data are being shared with the public so that machine learners like myself can validate their predictors they build on those lab data on real clinical materials. But there are many biomarkers or there are many therapies for which this is not possible because the data are not being shared with the community. So a lot of work to be done there. We have a lot of predictors of drug response, but very few actually been validated with patient materials, which is usually important.
So what kind of biomarkers do you have at the moment?
So most of the biomarkers we use in the clinic are DNA-based. Those are mutations or gene fusions, and they're usually single features, a single type of mutation or a bunch of variants. But we don't really have very complex multi-gene predictors. There are a few. In breast cancer, for instance, we have the Oncotype DX, where multiple gene expressions have been combined to predict breast cancer response to, let's say, tamoxifen or chemotherapy. But this is more the exception than the rule. So now we're really moving forward to multivariable biomarkers. So we use multiple genes to predict the response to drugs, recognizing the complexity of the mechanism of action. Mostly using RNA. DNA kind of showed a bit its limit in terms of predictive value. So now we're looking at other modalities that are either being implemented in clinic now, which is the case for RNA sequencing, or other technologies that are soon to be implemented in the clinic, such as chromatin accessibility via ATAC-seq, for instance, or methylation. So that's the kind of technologies we use to collect the information that we need to build those deep neural networks and build those predictors of drug response. Not all those modalities are ready in the clinic yet, but we mostly focus on the modalities that will soon be implemented in the clinic. So trials are being organized now with those AI models in mind. Most of them are still in the research settings. And there is also the problem of regulation. AI is a very special technology in a way that it evolves very, very fast. you can imagine that even over the course of a trial, you could use the data being generated to update the model in real time, which obviously you need an adaptive clinical trial design. It's fairly complicated, but at the end of the trial, it would be somewhat inefficient to ignore that data. And most researchers would say, well, let's use that new data to make our model better. But then from a regulatory perspective, it becomes a big challenge because this model will evolve over time very, very quickly. So re-evaluating the new versions of the model is really something that people are looking into that now. When the clinician is going to have that in their hands for standard practice, that is going to be also an issue. Which version of the model have you been using? How do you understand the difference between version 1 and version 2? Is it really better for the kind of disease that you're going to treat? Was the update of the model specifically for the kind of patients you're seeing? So we're still going to be empowering the clinicians. There is absolutely no question that the clinician will have their hands on the steering wheel, but AI will help integrating way more data that it's humanly possible.
To sum it up, what are the advantages and also the limitations of using AI for new biomarkers?
Okay, so for the moment we are working on what we call narrow artificial intelligence, which is we build model for a very specific task or a set of specific tasks. Now AI doesn't really have, at least now, an understanding on how the world works from a physical perspective, from a human societal perspective. So that's where the human really shine. The clinicians seeing the patients have a lot more information that will ever be accessible, not ever, but that is accessible to the AI model now. It could be a time where we collect so much data about our lifestyle and our social interactions that we can feed the artificial intelligence model with that. And of course, there is a lot of privacy issue with that concept. But for the moment, the clinicians still has a lot of extra information that the AI models won't have. So it's really going to be this collaboration between the AI models that can dive deep into a certain type of data. It could be very high dimensional, like sequencing data, for instance, or even clinical data to some extent, with the clinicians who see the patients face to face and understand the preferences of the patient, the performance status of the patient. Will that patient be able to really handle that line of therapy, quality of life? So there are a lot of questions that will ultimately still be the clinician's decision, at least for now. I think it's going to take probably decades before the AI models are being fed with enough information that they can handle all those cases. So the clinician will still be the last line of decision, and the interaction with the patient will be very, very important, despite the most accurate AI prediction you can derive from those high-dimensional clinical and sequencing data.
Is there anything you want to add?
I like this analogy. So there is this very famous quote saying that in radiology, for instance, where we look at images and we have to segment tumors and organs, radiologists not using AI will be replaced by radiologists using AI. And this is still very, very true. So people saying that AI will replace clinicians are dead wrong in most applications. When the decision is sufficiently complex, the humans will still have their hands on the wheel. Now, I really like this parallel with other fields such as aviation, where you have different levels of autonomy, level 1, 2, 3, 4, 5, depending on how much the AI is able to take decision and control the effect of that decision. So would you step on a plane that doesn't have an autopilot system? Probably not. And very soon we'll have patients asking AI-assisted clinicians. They will have more trust in clinicians using AI because they know that they will be able to integrate probably way more information than clinicians not using AI. So this is really, I think, the way we have to educate clinicians and patients regarding those AI technology. They're really here to assist. They're really here to augment human intelligence. They're not here to replace, for most cases, the work of health practitioners.
Thank you very much.
You're welcome.